All right. Good evening, everybody. Thank you for coming to the Commons for tonight's Rock Talk. So I'm Dave Buck. I'm the Associate Director here at the lab. Our Executive Director, Sarah Morris, is off the island for a bit of this week, but she's been to inform with ours, which is here. Um, so this is our fifth Rock Talk of this 2025 season. No, right? Did a great one so far. Um, these Rock Talks are a great opportunity for all of us to kind of come together under one roof. You know, during the day, you all are scattered for wind, doing stuff in class, doing stuff in labs. And these seminars provide a great opportunity for all of us to come together and hear from a professional in the field. I have Nick Gidmark with us. For those of you that are joining us online or who may be new to the lab, Shoals Marine Lab is the largest and oldest marine lab that's solely dedicated to undergraduate teaching and research. And the lab is jointly operated by the University of New Hampshire and Cornell University. So in addition to those of you that are with us remotely tonight, you should know that we've got a handful of folks here on campus with us. First, we have our field animal behavior class where we all give a shout out. Yeah, all right. We also have our anatomy and function of marine vertebrates class. In addition, thank you. In addition to our two classes, we also have our Shoals undergraduate research group, who is, starting their third week of a 10-week undergraduate research experience. There you go. Well done. We also have a small but significant team in college who study unique geologic features. Come on, Smith College. All right, perfect. So that's who's here on island. Folks at home are going for this talk. So the format for tonight's talk is 45 minutes, give or take, followed by a 15-minute Q&A. For those of you that are online, please use the Q&A function on your Zoom link to post questions, and we'll address them after the talk. The audience, I'll walk around, ask them room and folks online, questions. So with all of that, so this evening's speaker is Dr. Nick Gidmark. Nick is an associate professor at Knox College. He earned his BS in conservation biology from the University of Minnesota and a PhD in ecology and evolutionary biology. After finishing his PhD in 2012, he worked on a handful of both NSF and funded postdoctoral fellowship at Brown University, at Harvard Labs, also at the University of Chicago. He joined the faculty at Knox in 2012 and then recently earned tenure too. So congratulations to that. Awesome. As you will see both tonight and then for those of us that are here on island, the next couple of months, it is a tremendous. Yeah. Different station. Nick is a tremendously enthusiastic and supportive graduate teacher and mentor. He's the only person that I know who has an email tag optimist that you see on the screen. You'll quickly find that things infection undergraduate students. It's a very limited education officer. It's also evident from coming here, he dedicated at least 2 weeks of his life every summer for the past 20 years, I think. Come here to Shoals our educational mission. So in addition to his passion for teaching and mentoring, he's also his lab at Knox College. His studies of musculoskeletal biomechanics, focusing on food capture processing. So I a little bit about that tonight. And his research has kind of taken him throughout the vertebrate world, everything from swines to fishes, to sharks and salmon. So without further ado, Please join me in welcoming Nick. He's going to talk to us from his perspective about the importance of animal variation. So without further ado, welcome to the Rock Talk page.
Thanks for that introduction and for having me here. I've been on that side of the podium like many dozens of times. Well, every side of that podium. But the reason that I'm able to be here, in all honesty, is really because of the people that I've got to work with. So I want to actually start with an acknowledgement of this smattering of incredible scholars that I've had the honor of working with for the past few years. I know my name is on the door, but big notes that's the of the lab. I'm the stats. So I want to talk to you today about not only food capture and how we deal with things in general, but I want to talk about some of the complexities of the different issues that are involved in the importance of both anatomy and motion. So I'm going to start with a video of a friend of mine, this is Zach. Actually, I will pull up a friend now. I'm going to eat a burrito for you. A little dance there. And as you're watching him, I want you to think about all of the different pieces that have to work together. This symphony that's put on by all of these different individual members of an orchestra in order to make food able to be processed. So we've got hard tissues like bone, like dentine and enamel. We've got soft tissues like ligaments, muscles, hip tissues, and all of those have to work together in a perfect synchronous way. If his left molar was 3 inches taller, none of that would work. Right? If his lips couldn't actually stretch around his feet, none of it would work. You actually have to have all these individuals that are talking accurately to each other and that are working in concert to make this thing called food capture and processing. I was sitting in my office a year or two ago with one of my students, and we were she was writing a talk for a presentation she was giving, and she came with this title, and I'm going to embarrass myself with her. And Alyssa said, you know, we're interested in all these different aspects of form. So we should say on the relevance of animal variation. And I was like, that's pretty bold, right? But it's true. All of these things, we have to understand how animals vary in order to understand how they work and in order to understand how different organisms function differently. We're going to have kind of a case study of mammals today. I know it's marine lab. I like marine biology. We're going to switch a little bit to marine mammals, but I'll talk a little bit about the marine in a second. But the point here is that mammals, even that small group, the relatively small clade from vertebrates and certainly a tiny fraction of life on Earth, are hugely diverse. We have all these different species that are eating all these different things in all these different environments with all these different shapes. And it's astounding, even within mammals, how much variation there is in there. So it's not just, here's the way that a mammal chews. Here's the way that this specific mammal chews when it's eating this kind of food in this kind of environment. All of those specifics are huge. And it's not just that motion is a huge part of it as well. There's two mammals here that they're both herbivores. The kangaroo is chewing in a really funny way where they actually push their teeth forward. They slide their jaws forward and back, which is totally different from the way that you chew, which is moving the jaws side to side. Just that difference between going like this and going like this is a huge difference. Changes what kind of muscles are being used, what kind of soft tissues are being used, changes what the food is experiencing. Motion is hugely important. And it's so important that even way back in 1887, Simon Leibniz set out to sort of catalog how animals focus mostly on mammals. But interestingly, I want to note that bottom right, he published an 11-volume set in 1887, back when you had to set the printing letters in the thing and push them. Crazy to think about how much work went into just publication of this work, and it's hugely important because he can tell us how basically all of the mammals through essentially stop animation and imagery. So he was even able to recognize way back then, we've only learned more over time, how important, how complex, how an animal works. Fast forward a couple hundred years or 50 years, and we've developed this thing called an X-ray. So lets you see inside, vision inside of an animal. And it turns out this was another revolution in understanding how anatomy moves and the importance of animal motion. This happens to be from a study from the lab that I was a part of as a PhD student, looking at how ribs move and function in ventilating the lungs. So we can talk about movement, whether we're talking about jaws or other body parts, limbs, all that's going to matter. And the way that that animal moves is going to tell us a ton about how that animal works. Here's our stint for just a moment of marine biology. This is a paper that I published. But I was looking at sand lances, which are these little eel-like fish that live around here. They also have to live on the West Coast, and they dive down and they swim in sand, swim in water, and then they also both float under the sand. And we used our X-ray imagery to look at the motions involved in swimming in those two environments. And up here, so up here in this top left panel, you can see an animal flopping around the body of that eel-like thing. Right below it is a trace of line looking at how that animal moves over time. And because water flows, those undulations are flowing all over them. That tells you that that thing that animal is pushing through is. If we look at that same animal swimming around under the sand, it looks like that upper right image there. Here's the nose there, the rest of the body kind of in a big head shape. And if we look at that over time, those undulations, those pushes don't ever flow. They don't ever move because the sand is supporting it. Turns out that's completely biomechanically different. Same animal, two different environments, moves differently. And we can learn things about the mechanics of the environment based solely on how they move. We've had another advancement in studying animal locomotion with this thing called X-ray reconstruction of moving morphology. I was on the development team for this technique when I was with you. And basically what we do there is we take one extra level. We have X-ray video cameras. Here are some X-ray video cameras that were pointing at an animal doing a thing. In this case, this is data. A friend of mine's paper looking at the way legs move birds. And we take those two X-ray videos where we can reconstruct those from two. We then take that bit and we have little tiny markers. They're red dots here. Those are little metal spheres that were surgically implanted before we take those things. We can then track that motion in 3D. And then we can take a CT scan of that same individual and find where those markers are in the bone very precisely. So now we get this truly precise 3D motion combined with this truly precise 3D anatomy. We combine those into a reconstruction, we call it, where we're actually looking at that animal, the bones of that animal moving. Video. So this is an example of pig data set. Sort of foray, little mini pig there, month pig, the X-ray video there in the screen in the back now. The animal's like totally different. Then we put those things together in this really complicated program called Autodesk Maya, which is an animation program Pixar uses. So we use this sort of, it's made to make things look cool, but we've taken it and co-opted it for a scientific precise. And so what we can do with those data are really amazing. We can now take truly 3D motions of skeletal elements and reconstruct what the locations those have for different locations. Here's one example. That big data set ended in a really interesting finding that pigs are actually chewing with both sides of their mouth at the same time. So the jaw actually elevates and then moves sideways and then goes down, elevates back up on that side and moves back. And we tracked the tuft of the teeth. That's those little dotted lines on the screen. And we show that there's actually chewing going on. breakdown going on both sides of the jaw. That's totally different from the way you chew, where you take a bite and you chew on it on one side, maybe you eat the other side. But these are effectively pigs are feeding like pigs. They're feeding with both sides of their mouth at the same time. That's how they're able to feed. So, a couple more cool findings that we got out of this work was we did this same study on macaque monkey. This was at work at Chicago, and we were trying to understand the mechanics of how an animal swallows. We looked at a bunch of different individual muscles. I'm not going to go into the details of that here, but we published a series of papers, and we have one in review right now, that show that this is a great model system for understanding chewing and swallowing the food. So now all of a sudden there's like some clinical relevance in this work that would otherwise be. We also had a super cool paper come out looking at the tongue and curling of the tongue. We showed that the tongue is, when it's pushing through one side of the mouth, it's not the tip of the tongue. The tip of the tongue curls under, and then the big wide portion of the tongue, squeezing it, the food around in the mouth. So this was actually a way to not only crack bones, but also crack soft tissue as well. So put some of these marks in the tongue. So now we can crack soft tissue, crack hard tissue. We can understand how these different elements are moving in space and explore other things. Like, for example, let's measure a problem. So we took possums and we were interested in not only the jaw and skull, but also the bone called the hyoid, the back of the throat, that supports the base of the tongue. And one of the things that we were able to show was just based on the posture of it, the tongue and the tongue base are in a different position, which, as you might remember in the other previous slide, means that there's probably differences as well. Another random tidbit from the lab that came out really recently was a paper from my co-instructor Emily, who was showing, actually quantifying the front-back motion that happens in fat, exactly like that front-back motion. Only we're actually able to quantify it. So I know what you're thinking. You're like, okay, you just found like 42,000 different little random information. Who cares? What are we going to do with these data that's actually useful for a greater purpose than just saying that a possible? Okay. And one of the sort of directions that my lab has taken over the last few years is that we've been looking at this really cool joint that's only in mammals called the acromandibular joint. In most other species, the jaw joint is made up of different bones. Oddly enough, the lower jaw is made up of several bones. So you have a different articulation of lower jaw with this bone. In the evolution of mammals, most of those extra bones went away or to other parts of the body. So we have just a single lower jaw, what we call the mandible, down at the bottom, articulating with the temporal bone of the region of the skull. We have this temporal mandibular joint, relatively across mammals. And so now we can start to understand some important aspects of variation. That joint is very complicated. Any of the members of our lab can tell you about how there's just 1,000,002 different aspects of that bone thing that we aren't explored. This is a figure that shows a few of them. There are ligaments, an articular disc with sort of the cushion or the feedback of the temporomandibular joint that helps to provide support. There's ligaments that attach to that disc. There's ligaments that attach to the mandible, to the skull. Lots of different pieces of that joint itself, not to mention that it's driven by muscle and something and other parts. What's more important, we've been talking about motion. Motion is going to be important. Turns out that the mandible moves in every possible way. So we'll get into it a little bit in the slide, but there's ways that we could slide different directions, there's ways to rotate in different directions. And it turns out we've seen most of those different types of motion of the lower jaw. So in almost everything that we look at. So again, on to why it matters. Turns out that that joint gets screwed up a lot. In fact, probably 30 people in this room have some disorder of that temporomandibular. Kind of wild to think about. And some people don't know it. Some people have clicking or range motion or pain or locked jaw, or sometimes the jaw will articulate. And that's a super important public health problem because it's like the only disease that I know of that 50% of the population. amount of tissue in this world that is all related to a single pathology. Can present in lots of different ways, pain, range of motion, but we call that a temporal mandibular disorder, or TMD. And so what our lab has been focused on for the last few years, now we're focused on the clinical relevance of this joint and how it varies across. One important implication of this disease is that it can limit range of motion. So the pink dotted lines there are looking at the number of degrees that's working that a jaw can go through if it's pathological, have the TMD, and the green is the controllers that help. We have a restricted range of motion. I could put up 2 dots like that are demonstrating different pathological implications of TMD, but this is the easiest one to graph one, getting less motion as we move the jaw. Problem with diseases in humans is that they're really hard. So there's a group in Switzerland who's developed this seemingly medieval torture device that they attach to the lower and upper jaw, and they are tracking motion of one of these relative to another. And I feel like we can all agree that that person is probably not moving their jaw when they have a retainer like that. They're not moving their jaw in it. So we're sort of restricted to either these really invasive materials or approaches, or sort of modeling approaches and things like that. It's really hard to get data about how a regular person. Because of that complexity, we often use model clinical and biomedical research. Turns out this smattering of organisms is really common as model organisms for studying jaw joint problems. Each of them for sort of different reasons, whether it's because we understand their neurobiology or biomechanically they're similar to us, or they chew in the same motion and things like that. But short version of it is that they're all species that are used commonly to study problems with the jaw joint. And then those studies are then transferred or translated, as we say, into, say, surgical approaches or other kinds of treatments in humans in particular. So what that relies on is that the findings in this species actually mirror the problem in a human. That's right. I don't know how good your skeletal biology is. Look at that mouse, right? Looks nothing like it. Yes, it has a temporomandibular joint, sure, but it has a totally different face. The muscles are oriented completely different. It's chewing with a different motion. I already talked about that with the front and back motion of the jaws. So there's a lot of variation. And so when Alyssa makes that title that says on the clinical relevance of animal variation, she's serious. Like, we need to look at all of the different aspects of how these animals move in order to understand how translatable that research is into actual phenomena. I'm going to talk through 3 specific studies of a whole myriad from our lab that we pulled out. A couple of these faces you might recognize on Highland. Again, I'm just jazz hands. This is all their work. I'm just kind of hurting, so trying to hold the work together in the lab. But I'm going to talk about a little bit about whether or not we can use XROM, whether or not it's precise enough to actually study what we'd like to study. We're going to talk a little bit about motion, and we're also going to look at how the surfaces of the jaw joint work. And these are the students who kind of pioneered those. So the first one that I'm going to talk about is Emily McFarland. She was interested in how precisely this technique, this approach of X-ray video, CT scan, how it works, how precisely we can read stuff. And so, being the very kind of animal lover that Emily is, she took a bunch of animals, we happened to think that, and then she took those heads and vacuum sealed them, and then she put them in a. She then took those animals, marked with the same markers in them, and moved them around with the X-ray videos. Then used that whole workflow to reconstruct how the jaw is moving relative to the skull. Dead animal, throwing them back to the field. Completely unable. Here's what you got. So it's kind of weird, right? Like, it's not moving. So that's initially you might be like, Nick, why? Really? What that jitter is that you're seeing on the lower jaw, that's the noise in this technique. That's the imprecision implicit in going through that ortho and reconstruct. What that tells us is how precisely we can reconstruct pathological versus individuals. Here's some of her data. The solid bars on these graphs are motions that are in healthy individuals. The open bars are motions that are happening in pathological individuals. And these are from a variety of different studies. They're on the x-axis, or shown along the bottom. And the pink error bars are the standard deviation that was reported in that study. And so, for example, if we look at the left one, that's sliding front and back, how many millimeters of sliding happens in healthy versus pathological individual. And the standard deviation, those pink error bars, they're overlapping. So this study published papers saying, here's how they move pathologically versus healthy in non-statistically significant. Which is kind of funny, but apparently we can do that in the literature. So we did. The green error bars, though, is the range of the imprecision in MLAC. Range, not standard deviation. If I were to publish standard deviation, I would put standard deviation on there, those error bars would be imperceptible. What that means is when other approaches are reconstructing motion, they don't have enough precision to reconstruct healthy versus pathological. If we were to take our technique and have the same variation, but the precision of this X-rom technique, we would clearly and cleanly be able to associate those two pathological versus health. We have way better precision than other clinical approaches. Kind of cool. So we also had the lucky fortune that a lot of these species that are used in as models in temporomandibular disorders have been already X-rom. That's fine. We already have X-rom data existing in the literature, some of which was published by our lab, which was published by associated labs, but we have access to all. So now the cool thing is we have this playground of 3D motion, all these different species that were used for other studies, how neural control and things like that. But we can use those to ask new questions about the world. The way we do this, or one way we do this, is that we look at how much motion is happening in different perfections. So we attach these 3D apps to the bones. This is all again on the computer, it's all virtual. And we look at how the lower jaw moves relative. We can do that in two different ways. One way is in translation, that is basically sliding. So if I had to use the trace of this one. We can look at sliding of that jaw front and back. This is being a little bit dramatic here, but you can slide front to back. You can slide side to side. You can slide up and down. Right? Those are three separate motions. We can quantify all of those individually. We also can quantify rotation about any of those. So we could take that skull and look at how much rotation happens long as. We could look at yaw, which is that side-to-side motion that you do. Or we could look at pitch, the normal, which is the normal sort of up and down motion. Again, we can quantify each of those motions independent of one another. So now we have 6 possible motions that we can look at, and we can compare them across. And so what we can do then is we can say, okay, when that animal is chewing, what are the motions that are principally involved in a translated to jaw form? And so when Non-Joy in our lab was interested in these motions, he asked the research question, what is the variation in the range of motion that happens during normal chewing in these 3 species? And one of the things that we found is that translation, that is sliding of those jaws, is first of all way higher in a rat. That is sliding the jaw all over the place. Part of that because jaw joints, like people. But there's a ton of sliding front to back and side to side that's going on. Way more sliding than is happening in other species. And that's sliding of the molar, just that. And then she also tracked the sliding of the jaw joint itself, which is obviously and we showed that that sliding is actually the condyle itself, all of that ball and socket sliding around the socket. Graph was super weird, very open socket. And so this animal is sliding its jaw all over this in a way that neither of apes nor monkeys, presumably not people, do. Okay, now we have we know that probably, at least in terms of motion, rats are not actually that good of a model organism for chewing. We're trying to translate those surgical approaches or those other in also, one of the other things we found is that rotation is again much higher in a rat than it is in a pig or a monkey. There's more of that rotation about all three of those different species. And so again, it's like rats are moving a ton in a way that other species don't. So take-home messages from Non were that there's primarily front to back sliding, but there's also sliding in other directions, and that there's just so much more movement. So I also mentioned there was electric soft tissues are super important. Obviously, things like muscles are important for driving motion of these of the jaw itself. Bone is also going to sit there. I'm going to do a muscle. Not only is there a ton of muscle involved in moving the jaw, there's also a ton of soft tissue, other soft tissue. There's ligament and tendons that are there. It's kind of a zoom in. If I were to take a slice from this side of the face of that and grab it, we'd see that this is. So here's this CO label is the condyle, the ball of that ball. So okay, here it's this thing that articulates with the skull. This is the fossa right here, the socket of the ball of socket. But there's also stuff in there. So there's the big, like I said, beanbag chair cushion made of fibrocartilage. They're padding the lower jaw joint. Lots of different individual pieces of soft tissue. And one of the things that we started to focus on is that cartilaginous, fibrocartilaginous disc. We think it's super important. There's a lot of literature that shows it's vital to cushioning and also just normal motion. It turns out that when you have problems with that, that's what causes things like osteoporosis and osteoarthritis. Lots of huge problems with the jaw joint stem back to problems that have shown up as part of that disc problem. We have this specific way that we like to study the jaw, the functioning of that disc. These were developed by that group that had that weird retainer that making them all over the face. And that technique is called identifying the stress field, which is essentially the nearest area of the fossa and the condyle of the ball. And we can look at how that moves over time. This is from a paper out of that group. And what you're looking at here, great bugs, but they're representing the condyle of the condyle, that ball, that's in the dark gray. You're looking essentially through the fossa, that socket, to see that condyle. Where that red area is the stress field, and where that dot in the middle of that red area is, that's the midpoint where we think the most stress is going on in that and we've got a lot of data that from that lab group that shows that location of that stress field and how it moves is deeply important to the functioning of that jaw. And when there's problems with it, when there's pathology, it's going to move in a different way. And so this is the kind of avenue of the lab that Alyssa Springer was championing for many, many years. And we're sad to say, back, please. Come back, please. That's actually a picture of 2 years ago with here at Gold. And so Alyssa asked the question, can we use this really cool software animation program that's really complicated and really honestly devoid of any logic known to man? And can we sort of use the tools there to try to reconstruct an analog of the space? And so years later, we did it. Which was if you know anything about coding via like respect. It's like a serious programming language. But she was able to, with some collaborators, identify a technique, a workflow to reconstruct what the distance of that socket from that ball is at any instant in time and across the whole landscape of that joint surface. So she's essentially reconstructing the topology and the distance between of that joint space. One of her metrics was to look at where that stress field is positioned in space, whether it's positioned toward the front of the socket or the back of the socket. And I'll summarize 4 years worth of working here and say that it does change across the body. Where that stress field is in a different place in different seasons. So we have three individual monkeys, 3 individual pigs, and 2 bats that are whose data we reconstructed to approach this technique. And we showed that front to back, the pigs typically tend to have that stress field very front toward the nose, and monkeys tend to have it very far toward the back. In very different places. Rats are kind of in the middle, so there's variation in where that stress field is located, depending on what species. And this makes sense, actually, if you look at the morphology. So pigs tend to have this really weird eminence. Monkeys have this really big eminence, this really big protuberance of the socket right on the posterior side. It's kind of gripping the back of the ball, of the condyle. This PG label right here. And so we think that's why that stress field is actually located posteriorly in that. In a pig, there's no posterior eminence, but there's an anterior eminence. There's a grippy thing on the front of that socket, which then makes that stress field point be in the front. So now we have, first of all, done it. We have shown that there is a way, she developed a way out there, but we have a way now that we can look at the mechanics of that joint and actually demonstrate it's different in different species. And so if you're looking at a pig, you may not be looking at the same mechanical stress-based interaction, mechanical system that you would be if you were looking at a monkey. And so not only does sort of motion certainly matter, but really it's that morphology, the anatomy of that matter more than how they move, at least in terms of how the stress field. Turns out, like I said, I've had the honor of working with a whole ton of different people. Here's a smattering of it more. I'm not going to explain exactly how these processes work. It's going to take too much time, but the way that you should look at these is the different colors are different. And when an animal is moving, all of the different orange dots are how that animal functions over their motion. And the different colors are different anatomy. And these 3 students looked at different aspects of how the jaw works, or how the jaw functions, I should say. That top one, Saxon Alvarez, is looking at the spaces in the joint in every direction, how much space that joint has, that ball has to sort of play around in that socket. Amira Sadiq is in the upper right. She was looking at the way that the lengths of the muscles and torques change across the rope. So is a muscle set up in a different orientation? And Rosalie Ross, down in the bottom right, was looking at the ligaments that are kind of holding that skull jaw together. And what's interesting, if you look at these plots, and they're kind of tight on this, I'm sorry, but what you can see is that all of the orange dots are clustered together. All of the green dots are clustered together somewhere else. All of the orange, purple dots are clustered in a different way. What that means is that, yes, movement is changing where that animal, how that animal works, what its biomechanical setup is, but there's a much bigger difference between species than there is within. So again, that brings us back to this kind of classic conversation that we often have in the lab where we say motion is so important, it really matters how somebody moves. I set this whole talk up to say motion is super, super, super important, but then those in the lab would say, well, maybe, because morphology is going to be even more. So it's like bringing us back to our roots as anatomists, that structure is going to be really the foundation of differences in mechanics and the function. And that's what we should be paying attention to. That's what we should be thinking about. Sure, motion is important, but that anatomical variation is hugely important to functioning of things like the lower jaw in mammals. And so as we're trying to evaluate whether or not the clinical treatment approaches will work, we probably should use something that resembles, looks like, functions in the same way as human instead of one that looks like. I want to say again a huge thanks to the folks that make this whole make all of the data work and also insane and crazy. All of them, and I've 41 minutes.
Congratulations. Thank you very much, Nick. We've got some time for questions.
Thanks so much, so it seems like there's so much variation in all of these different aspects among the 6 standards. And so I'm curious if y'all use that lab to figure out like which animals come out on top as far as closest to human.
We'd love that. That would be wonderful. I think that would be totally a gold standard. That lab is in Switzerland. People in general, and so I'm not sure that they would be excited about sharing that approach with us. The other thing that we've been thinking about is that in some ways, replicating that technique on these other animals, first of all, it'd be really hard. Freak out. But also, that may not be the gold standard of comparison. We could get some humans to volunteer to have markers surgically implanted in their skulls and then do that technique on them. That would be the gold standard because that would be more precise, as we showed with that first set of data of evolution. That would be significantly more precise and would actually be comparable to other things. One thing that we could do, if somebody were to decide to let us, might be to develop some kind of implants where we could put markers in those dentures and still get them in place and still be able to reconstruct everything and avoid drilling into people's jaws. I don't really want to do. So that you're right though, that comparison is important.
Thank you for your talk. I was curious, and you might not know the answer to this, but why did TMJ specifically evolve in mammals? And was it diet related, or did it evolve as a result of other evolutionary changes in mammals? Yes, I wish.
I'm into it. Yeah, no, great question. We don't know the answer to that. And actually, honestly, what we've been spending not in our lab at all, but there are many labs who are interested in that question and who are studying, especially on the record, to look at that pattern of tape. And it's really weird because those bones, a couple of those bones that were there in birds, fishes, and everything, a couple of those migrated into your inner ear. So your sleepy, like some of those bones, like inner ear bones, are actually super weird. And we think for a while that there may have been separate jaw joints, where there's a jaw joint between the quadrant and the articular joint of everybody else and the TMJ. Presumably diet related, but mammals are really diverse, so it's kind of hard to know that those mammals are kind of true-looking things. But we don't know what was the
I actually have 2 questions. My first is if 50% of people have
bingo. Nice.
What would you recommend to do going forward? Like, if they're not going to start speaking?
I have a friend, right?
Yeah.
She
I
has
know
to.
that.
Yeah. Yeah, there's actually a ton of doctors out there who work specifically on PMD. There's also like there's a ton of research that goes into it. So the one of the national Institutes of health, the NIPR, the National Research Extension and Facial Research, like compared to their budget. There and some of that funding model organisms, some of that funding all kinds of other research. So there's a very active base of research, yeah, still critical. Yeah.
The other question was, do you ever go back where you're going on the so you will be looking at how
So the big study that has republished individuals has a horse individual that we study about. The data didn't make sense, and we put one sentence in a paper that said, and like just left it. So that's kind of the next step would be to look at that species that we know was moving in a whacked out way, and then try to say, we use some examination of the CT scan, if there's osteoarthritis and some other kind of pathological bone growth that might suggest some kind of pathology there. But yeah, I mean, it'd be great to get. We actually have data. We were able to get Dever material. So we're trying to do all the people, but yeah, great.
how common are issues with the temporal language with other organisms? I know humans, we have had like a narrow jaw. All
And okay.
yeah, at or
Yeah.
do other animals that have had this kind of not off their jaw that comes up with
So I can't speak to all things. I do know that Alyssa did some research on this actually told a couple years ago looking at the jaw joint with patients, and we found like literature stuff about uh,
Temporomandibular disorders, and specifically it was looking at like abnormal bone growth, most often what they had. Wasn't super prevalent in patients. But it was about 40% the specimen that they had found, and these were naturally deceased animals in the pinnipeds actually had some form of abnormal bone growth or osteoarthritis in the jaw joint. Kind of reasoning there was that they're doing a little bit more mechanical processing than certification might be. So yeah, it's definitely out there, exists in the wild in other species. I don't know all animals, I'm sorry.
So you've been talking a little bit about how you would have to something. Is there any other way to get that data without doing the implants?
So you have to have, great question. You have to have some kind of cross reference between this scan and the video. And I was part of a study that was trying to do a similar thing. And that's pretty complicated with most joints. But it turns out the knee of humans is really far out from the body is pretty biomechanically simple, and it has a lot of useful shape. And so we toyed around with writing a lot of code that would try to virtually create different positions of the end of the femur and the proximal end of the tibia, and fake X-rays that would shoot through it and look at the image that would have been produced had they been in that position and compare it to the actual video, which is like really computationally intensive, but it doesn't. And actually, we had a couple of papers about, but it's and so the guy who headed that up, Dave Merrill, did his PhD on it, and took him to do like 7. And so from the standpoint of, can you do it? Probably, but how labor intensive would it be, even with the addition of AI stuff? Is done. And we had a grant in with Chicago into NSF to actually integrate AI into that approach, the smart register things. But I'm glad you waited until. Gotta get steps in. Keeping you young, Dave.
So my question is, could like the rat with their open jaw mechanism with logical variation be an evolutionary result of having problems? Like maybe, maybe their problem with their jaw when it's tight had evolutionary actually failed them, so that the loosening of their jaw could be an evolutionary result of success and survival.
That's an interesting thought. I don't know what the prevalence of osteoarthritis and pathology is in normal rats. They are, what's cool about them and why they're not is that we have genetic programs where we can essentially induce osteoarthritis and other types of beginner rats. Kind of the opposite of what you're asking. But I mean, I think it could stand to reason, because something like a pinniped, which these are available if you check them out. If you look at the fossa of a pinniped, it's actually really strange. It's like the opposite of a rat. And so from our N of 1 data point that Alyssa pointed out, that pinnipeds did a lot of TFD, that would go to your point. That would support your thought that having a jaw joint might make it less prevalent for you to have. So maybe.
Also, just
Well, I have a
Okay.
microphone.
just in general, rats, like, do rats have looser bone joint structure? I know they can fit in tiny spaces. They're very
Not really.
flexible, but not like,
No, most of their joints are pretty complicated.
okay.
Just kind of a general grant question. It's like a really thing, like you said, you're generating a ton of data, and this is a very cool innovative technology. Is there an effort to, or like, how do you make that open? Is it something that you can post that data anywhere? Are you creating products and tools? What's like that?
So so any publications that we bring out of that information, that data, we have to publish in a repository that's accessible. In fact, I happen to be involved in all of those studies, so I happen to have the data like on a drive anyway. But if I didn't, like legally based on the on journal regulation, they'd have to let you think. So everybody has access to that data. The kind of sticky piece is that the process of XROM, that workflow, takes a long time to learn. And so in some ways, it's like not really fair that it's not as accessible as it could be. People can download the raw data for free, probably take it, but whether or not they could actually do anything, make sense of it is another issue. And Beth Brainerd, who was my mentor at Brown and sort of the spearhead of the whole XROM process, she's been very proactive about trying to disseminate that as a tool. She teaches a workshop on it every year for no money. People can come and check it out. got a whole bunch of tutorial videos of it online, and the goal in that it's very much in the spirit of science, trying to increase accessibility. But it's still an uphill battle because of how. So we do it in our lab, but it's like somebody comes in and they spend there for a year, like just trying to navigate how to do one of these things, you know? And so it's only after they've been in the lab for more time are they actually able to do stuff like code, create workflows. And another one.
I was curious if you know it is going to affect TMD. Like, is there any relationship between those things?
So, yeah, so it's amazing how dynamic your jaw is. Wisdom teeth are actually evolutionarily, we think that the reason they were there is because when you lost a permanent tooth, you had teeth that would literally push the other ones back and forward, and your jaw will on its own remodel as a result of movement. And if you don't, it impacts wisdom teeth. That's what's going on. Those wisdom teeth are trying to push all the other teeth out of the way, and they're all in the way. No way around it. And so there's a huge amount of dynamism in the occlusal surfaces, well, root surfaces of the jaw. That shouldn't directly impact the mechanical environment of the TMJ itself, but if that causes weird like malocclusion or something where the teeth are not coming together right, so they're putting a shearing force or working on the jaw joint itself, that could induce TMD. So like one of the ways that TMD is induced surgically in like rabbits, for example, is that the researchers will actually like attach a piece of suture from like the skull to the jaw to make the jaw like wrench to the side, and that induces all of those TMDs that pathological bone growth. So no doubt problems in occlusion could indirectly end up causing TMD, and those could be caused by vision.
Got time for one question from the crowd.
Yay. Yay, yay.
So I've got a question about the moments. Now you talked a little bit about the anthem. I'm curious if there's any relationship that you might have been seeing associated with like the movement to larger temporalis. We see we also see in modern Gerber's family large masters that is a bit more anterior. Is there any relationship between that and some of the pathological things?
That's a great question. So I guess my best response to that is that there's not. So in non-mammal, there is no master right for that. And that master is a snake morph for mammal, but it doesn't have it. And I don't know of the so but the relative input of say master versus temporalis certainly is going to impact the balance of the whole sort of environment of that judgment. And the more you the more similar the animal that you're studying is to a human theory, that especially in terms of like that kind of ratio, the organ that it's would be. So yes, report. Do I know how?
Question.
Do I have no, no, I don't.
So Nick, thank you very, very much for taking the time for this tonight. Appreciate it. Seems like one of the mammals missing is study. Opportunity
Go.
to work here on it.
I'll just bring my extra for my work.
So with that, we'll end tonight's seminar. Quick note about next week, we're going to be fortunate to have Dr. Will Kimler, who's also in the house tonight, teaching FAB, will be providing us with a talk on the ecological network of island topic for life here at Dumbledore. So with that, thank you very much, Dr. Gidmark. Pleasure.